A very interesting day yesterday, and hopefully I can bring more joy to you today. All right. And so today I'm going to talk about new technologies from proteomics to multiomics that can potentially help advance your research. And in fact, in January next year, we are going to have operated this core facility for 25 years. So, time goes by quickly. So I want to showcase some of the success we have achieved, and hopefully with new technology we bring on board, we can help you to advance your research. Okay. All right. So I see Carolyn here. So this facility was established in 2000 as a joint effort between UMDNJ and then Rutgers Newark. It was spearheaded by Dr. Mike Matthews, then chair of biochemistry, and Frank Jordan, chair of Rutgers chemistry in Newark. And then they entrusted then junior faculty, now full professor Dr. Suzuki and Dr. Rami Farid, and who recruited me to come and operate this facility with one very simple mass spectrometer. I'm going to show you later. And the goal of this facility is to help expand NGM's mission to advance research innovation using state-of-the-art proteomics technology. And during the last 25 years, we have been able to accomplish quite a bit. So we have about 200 researchers that run over 20,000 samples. And each year we help about 25 PIs to apply for their federal grants. And among them, 80% of our users are Rutgers users, which aligns well with the goal of institutional support for the core to advance research at Rutgers. And this core, as the PI and multi-PI, we have been able to secure about $15 million of grant dollar to both buy new instruments and then get money to support researchers to use the technology. In addition, we also help other PIs to secure over $20 million of other grants to support their research. In total, we published over 140 papers. The reason why I can't give you a precise number is most PIs, they pay for fee for service, so we don't know how much of our data they actually use to publish their papers. But in any event, we published a lot of papers, have created big impact on some of the people's research here at Rutgers. So without good people, we can't deliver good data for you. So I just want to introduce the current team of very dedicated staff at the proteomics school. So Dr. Tong Liu, who is sitting here, she joined my facility in 2004, came from Dr. Suzuki's lab. And so right now she's the main mass spectrometry expert, performing a variety of LC-MS experiments and perform the advanced protein quantification analysis that I'm going to talk about later. And then Dr. Chen Sun joined the facility from Dr. Marco Zaben's lab. She also has a lot of experience in molecular biology. Her main function in the labs is to perform sample preparation, convert proteins to peptides, enrich phosphopeptides, and then give to Dr. Tong Liu for analysis. And coming early 2025, we have recruited a new PhD, Dr. Ariful Islam from Bangladesh. He got his PhD from Japan, and with a lot of experience in mass spec imaging, which I'm going to talk about as one of the new technologies we are going to introduce new service to at the facility. Okay. So as I mentioned before, we published over 140 papers. As you can see that we've been able to collaborate with a variety of people, publish many papers. Of course, the real impact is many of users are able to get data to publish their own paper, which is not counted here. So likely impact will be higher. And our collaborators, we collaborate with people across the world. And of course, most of our collaborators are within all the campuses of the Rutgers, different schools of Rutgers University. So during the last 25 years, we have been able to continually to update our equipment about up to a dozen also equipments. Most of the funding is from the competitive grants from NIH and NSF. And so I'm going to explain a little bit why we need to constantly keep buying expensive equipment and why that this is important to help you to accomplish more with your research. So Dr. Lazzarino want me to provide a very basic training about what is mass spec and why we need different kind of mass spec to do different jobs. So I'm just going to provide a very basic overview of mass spectrometry. So essentially, mass spec is a machine can measure the molecular weight of a molecule. And for most molecules, because it has unique chemical composition as seen in the amino acid table on the right, and because of different elemental composition for each amino acid, it has different molecular weight for the most part. So this is similar to counting a pile of coins, and then each coin has different size. Okay, and then by measuring the size as well as count relative amount, we can identify and quantify molecules. So this is the simplest concept of mass spec. But of course, in reality, when we are analyzing biomolecules, it's actually a bit more complex. So for example, many molecules share very similar mass, as shown in this table on the left. So in between the mass of 132 to 132 and 2, there are a group of molecules that share almost identical mass. Okay, so when this collection of molecules we observe in mass spec, they all seem to show the same mass of 180. So how do we distinguish different molecules share identical mass? Therefore, we need more advanced instrument to do that type of more detailed analysis. So one way to be able to analyze different molecules with identical mass is to separate molecules by HPLC. Okay, as you know, that HPLC can separate molecules with a variety of mechanisms by polarity, hydrophobicity, size, etc. So this is an example showing you, separation of molecules by HPLC. All those molecules have the same mass. Actually, in my other slide has the mass labeled. Oh, yes, it's here. Okay. So for example, on the left, all those molecules have the mass close to 169. And then on the right panel, those three sugar derivatives all have the mass of 259, for example. So in order to identify them individually, then we have to separate them by HPLC. So this leads to the development of a technology called LCMS. So when I first came to Rutgers, we have an instrument that cannot be coupled to a liquid chromatography. So later on, when we get more grants for new instruments, we were able to do LCMS. And Dr. Balafato was one of the earlier brave researchers trying our new instruments on those Q-top instruments, right? Remember? Great. All right. So another way to separate molecular complexity of the molecules share very similar or identical mass is called ion mobility. This is a technology have been recently commercialized in the last 5 years or so. And this is a technology that can separate molecules based on their shape within the mass spec. For example, these two peptides, one is more linear and one is more globular. When they are being presented in this ion mobility device, ion mobility can separate those molecules based on the sort of like an SDS page in the mass spec, based on the shape, and the molecules can traverse with different speed in the mass spec. So the lower panel shows you an example of the lipid with cis or trans conformation. They may have identical elemental composition just based on their stereochemical conformation. They may show up as different shape and then can be separated by ion mobility within the mass spec. So this is very critical if you're getting into the lipidomics analysis, try to identify high resolution lipid species that differ from each other, not by mass, but by structure. So a third way to address the issue, how to resolve molecules of the same mass, is to use this technology called MSMS. Essentially, tandem mass spectrometry, also called MSMS, is a combination of two different mass specs in one box. What it does is to use the first mass spec as a filter to isolate only one precursor molecule to be fragmented in a collision chamber, okay, to break down this yellow molecule into 3 smaller species, and the relative size of the 3 smaller species can then be detected by a second mass spec. So the question is, how do we use this transition from yellow precursor large molecule to a collection of 3 small molecules to identify the molecular structure? I'm going to use this panel showing you a fragmentation of a short peptide. And it just happens that peptides, when being presented in tandem mass spec, can be broken apart pretty readily at the peptide bound area. So the outcome of that fragmentation is from this peptide of 4 amino acids, you're going to get at least 6 different fragments, potentially more. And then they each contain a portion of this precursor. By comparing the molecular weight differences of those precursors, you can read the amino acid one by one, and from this pattern. Of course, nowadays with advanced bioinformatics, you don't have to do this manually. So with software, you can do this rather fast, and we can quantify tens of thousands of peptides within 2-hour time frame. So what does this mean to you? Why do we spend the last 25 years have to update our technology every 2 or 3 years? It's because of this phenomenon showing on the right, okay? So assuming the proteins that are present in the cell or tissue that have very wide dynamic range, and typically mass spec sequencing happens from the top, analyzing the most abundant proteins from the top, then gradually go down. And because the relative abundant proteins, let's say in the blood, has a lot of albumin and IgG, and mass spec spend a lot of time sequencing those peptides, it will not be able to have enough time to sequence the low abundant ones. So therefore, there's a lot of technology development, both with HPLC and mass spectrometry, trying to push this boundary, allow us to identify more lower abundant proteins. Okay, so that's a constant struggle, as well as an area with a lot of technology advancement. So with our current state-of-the-art instrument, we should be able to detect about 10,000 proteins within, let's say, HeLa cell digest as a reference point, and be able to identify and quantify about 20,000 phosphopeptides. Okay, of course, with a lot of those information, you're able to do more advanced bioinformatics analysis and be able to identify potential novel targets and map signaling pathways. Even with those kind of advancement, knowing that there's about at least 25,000 human genes, at least 25,000 protein species in a given cell, we are still only cover about less than half of the proteins in a cell. So there's still a lot of room for improvement. Okay. And actually, this is a small group, so if you have any questions, please raise your hand. And for people online, maybe we can answer your questions when we finish later. Yeah. Okay. So not only there's a lot of advancement in mass spectrometry, there's also a lot of advancement in sample prep and the chemistry in order to allow you to be able to quantify more proteins, analyze more samples in shorter amount of time. So one of the more recent development in the quantitative mass spec is to label proteins with something called a tandem mass tag. Those technology allow you to quantify cells from up to 35 conditions and combine them into one tube, be able to quantify more than 30 cellular conditions at the same time. How does this technology work? So we have let's say 10 different cell lines that cancer cells treated with different drugs, whatnot, or wild type mutant cell lines. And then we can extract the proteins, digest them into peptides. And the peptides from each sample can be labeled with a particular version of this so-called TMP tags. And then those labeled peptides can then be mixed together and be presented to an LC-MS for analysis. So with this kind of analysis, we're able to get a spectra like this on the right-hand side with this ACSM5 mouse peptide. The right panel with labels of B or Y ion is the way software to sequence the peptide with that puzzle picture I showed you earlier. And on the left end, low mass region below 200, when we amplify, we will see the quantification region at this lower panel, allow us to quantify peptides for this particular peptide with the yellow, green, or blue and red tags. The region represents the relative amount of the proteins from different cell lines that we run the peptides from. So with this information, we can generate the heat map as well as the volcano plot, allow you to quantify a lot of protein expression differences, develop hypotheses. So as mentioned before, right now the tag allows to quantify up to 35 samples. So this technology is a 20-year-old technology. I'm going to show you 20 years ago, we can only quantify 4 samples in one experiment. Now we can quantify over 30. So that takes 20 years to reach this point. So what if you have more than 30 samples? So recently there's another development in how we acquire data to allow a process called the DIA analysis. DIA stands for Data Independent Analysis. So typically when Dr. Balafato or Dr. Suzuki gave us a sample 20 years ago, the way we can perform mass spec analysis is with HPLC separation on the time scale. At any given time, there are many peptides being presented to the mass spec. Mass spec he can choose one peptide at a time to sequence. And because the instrument works very fast, so we can only analyze maybe up to 10 peptides to sequence them. As shown in this field of flowers, we can cherry pick one flower at a time to identify them. More recently, the faster mass spectrometry allows us to perform a new analysis called a DIA analysis, Data Independent Analysis. For this analysis, mass spec do not pick one molecule at a time to sequence. We actually sequence a group of molecules at the same time at any given retention time. And of course, the result is we're going to get a mixture of spectra for many peptides at the same time. And then with advanced bioinformatics analysis, it's able to pick specific peptides from this mixture of spectra and allow peptides to be identified and quantified. The benefit of the DIA analysis is more sensitive than DDA analysis, and also it can allow us to quantify hundreds or if not thousands of samples. This is actually very critical for clinical application because whenever you analyze patient samples, if you do a human, you have to analyze large N numbers to get significant results. So I'm going to show you how we use those advanced technology hardware as well as advanced chemistry to help you to answer biological questions. So this is a general diagram showing you that a lot of studies starts with genetic analysis. Gene sequencing is simple because it's static, it doesn't change for a given cell. And once you go from DNA to RNA, there's a lot of differential processing, and then to translation as well as post-translational modification. There's a lot of molecular diversity being produced. Even the same gene product can generate many different molecular species based on RNA splicing, proteolytic cleavage, post-translational modification. Of course, one of the most useful applications that we've been working with is protein-protein interaction. Identify dynamic protein complexes in different contexts. So protein complex or protein interaction, post-translational modification, they are dynamic, meaning that in one cell condition, you may have a modification, or you have certain protein complex. In another cellular condition, you may not have it. So it's important to perform this in a dynamic fashion, be able to compare different conditions. So the kind of service we do are mostly focusing on the right-hand side of this panel, okay? So I'm going to show you a few examples with people that you may recognize. So the first story I'm going to tell you is to study the mechanism of nerve growth factor or pro-nerve growth factor in the neuron growth as well as cell death. So nerve growth factor, as the name implied, can stimulate the growth of nerve, axons, and dendrites, for example. So the protein is synthesized as a large precursor. And typically, this precursor protein, the signaling protein, allow the peptides to be in the secretory pathway to be secreted out of the cell. But in order for the mature protein to carry out its nerve growth activity, it has to be cleaved of the pro sequence. And then only the blue sequence, when formed with proper disulfide bond, is able to activate the nerve growth function. So this is a canonical function of this NGF. And so we did a study with Dr. Ray Birch early on when we first set up the core facility. And more recently, we published a paper with Dr. Wilma Friedman at Rutgers, New York. She studies another version of this protein called a pro-NGF, contains both the yellow and the blue region. And it just happens that this bigger protein derived from the same gene product can kill cells, as opposed to the smaller version of the same gene product can promote nerve axonal growth. So I'm going to talk about the project Dr. Ray Birch first. It's simpler. So this paper was published in 2006. At that time, Ray was studying a protein called FKBP52, a chaperone protein. And he was collaborating with Johnson & Johnson, and Johnson & Johnson developed a drug called JNJ-460, okay? And what they find is that this red JNJ-460 molecule, when incubated with dorsal root ganglion cells, the ganglia, when you treat with the JNJ-460, it can grow neurite-like processes, okay? And then this is very similar to when you treat the same ganglia with NGF, the shorter version of the protein I mentioned. So the question at the time is, can we use proteomics to identify whether they have a different mechanism and leads to the axonal growth? So at that time, Tong just joined my lab for 2 years. We were very excited to try this iTrack method. iTrack is a method very similar to the TMT I mentioned, but at that time, as I mentioned before, it can quantify up to 4 different conditions at the same time. So by quantifying the protein expression from JNJ-460 treated ganglia versus the NGF treated ganglia, we're able to quantify which proteins are upregulated in different cell treatment conditions. So the panel on the left showing you that one of the protein called the troponin T is a structural protein. It's uniquely upregulated by J&J drug treatment. And on the left panel shows the increase, and on the right panel shows the sequence we're able to identify. On the other hand, the proteins we can find from the NGF treated cells, we find the activation of translation regulatory factor EIF5A, but it's not found in the J&J treated cells. And the tables in the middle section of this slide showing you the upregulated protein from J&J treated cells, the treated ganglia. It shows you most of the proteins involved in organogenesis. A lot of structural proteins seems to be upregulated in this J&J treated condition. By comparison, the protein upregulated by NGF, many of them are involved in protein synthesis, biosynthesis, suggesting that NGF differ from J&J drug by stimulating protein translation. On the other hand, the J&J drug seems to elevate the structural protein, perhaps by stabilizing those structural proteins, preventing it from proteolysis, from degradation. That's just it. But clearly, the protein pattern indicates there's a big difference in the mechanism of the neurite growth. So what about the second story with Dr. Friedman? This paper was just published just last month in Science Signaling. In fact, it made it into the cover of Science Signaling. So it's a very nice story. So Dr. Friedman has been studying how pro-NGF, the larger precursor protein, leads to cell death. And so what they find is that this process, first of all, can be regulated by something called a retrograde transport, meaning transport of signaling molecules from the axon, from the synapse, back to the body of the cell, where the nucleus are. Okay, as you know, the nerve cell has a body, which has a bigger part of the on the right, and they have a lot of processes with long arms that can talk to other cells. Okay, so what they find is that when you treat the axons with pro-NGF, you have increased tunnel staining, increased apoptosis by the tunnel staining in the middle panel. And then this process can be inhibited by a drug called ciliobrevin. It's a compound that can inhibit retrograde transport. So suggesting that there is something needs to be transported from the remote area back to the cell body for this apoptosis to happen. And then the lower panel represents the fragmentation of the axons. So upper panel is the cell death, lower panel is the morphology of the axons. So when the normal axons is treated with pro-NGF, as you can see, a lot of debris from the axons as well as the whole cell is left from the NGF treatment. Okay, but this process can be attenuated again with the retrograde transport inhibitor, suggesting by inhibiting retrograde transport in the axon, this process can be attenuated. So the next experiment they did is to see whether if the molecules that is being retrograde transported is related to new protein synthesis. So when they treat the same assay with cyclohexamide, and apparently cyclohexamide can attenuate the cell death process as well as the fragmentation of the axons, suggesting the new protein synthesis is required for this process for the pro-NGF to cause cell death. So that's when Dr. Friedman reached out to us, want us to help her identify newly synthesized proteins in the axons, allow them to build a hypothesis to find the mechanism of this very interesting phenomenon. So to do that, they are able to grow very clever assay by growing the primary neuron in this cassette device. In this cassette, the soma, the body of the cell, can stay on top. The axons can grow in a hole in the bottom. And so they are able to isolate the proteins from both the cell body and axons separately. And so that allows us to analyze the proteins only from axons and compare that with the proteins from the cell body. So that's one innovation. The second innovation is they want to identify newly synthesized proteins, not only all the proteins in the cell, because if you analyze all the proteins in those cells, you may dilute your newly synthesized protein signal. So for that, they use a new technology called OPP. This compound can mimic tyrosine tRNA. So wherever tyrosine is needed in the protein synthesis, this OPP compound can be used by the protein synthesis machinery. And the net result is lead to premature termination of the translation. And this compound also happens to have a biotin attached to it. So we are able to use click chemistry to isolate the proteins attached to these prematurely terminated proteins. And then the peptides derived from this affinity capture, we're able to perform TNT labeling as well as LC-MS/MS to quantify what kind of protein, newly synthesized proteins are uniquely present in their model. So with that, we are able to quantify a few hundred proteins that are newly synthesized in the axon. And by performing pathway analysis, many of those proteins, a lot of cell death related proteins are found in the pool of newly synthesized proteins. And then proteins involved in cell viability is reduced. But because we have hundreds of proteins, how to find more insights and filter those data? So we collaborated with Dr. Friedman's group by putting the data into ingenuity pathway analysis. And then in that analysis, they're able to provide something called upstream regulators. So because they're neuroscience scientists, immediately they recognize APP as a upstream regulator of some of this protein synthesis we discovered in proteomics. And as you know, APP is a precursor that can lead to the production of beta amyloids, which leads to Alzheimer's disease and some other problems involving cell death and dementia in people. So basically, from the proteomics data, they're able to determine they want to study how APP can play a role in this process. And of course, they verified the APP induction in the exon by Western blotting. So proteomics is one way to identify a hint, and you always need to validate what we find with alternative approach. So they did that, basically verify this upregulation of APP happens in the exon, but not in the soma, not in the cell body. So this is a very interesting find. And then what they did next is to validate their study by knocking out APP. So long story short, by knocking out APP, they're able to stop the apoptosis of the exon as well as the fragmentation of exon after pro-NGF treatment. So of course, in their paper, there's a lot more details in developing the story. And the take-home message for their study is, so when there is pro-NGF that binds to the P75 receptor, this binding leads to nascent protein synthesis and leads to the axonal elevation of APP. And APP from their downstream experiment seems to cause an increase in calcium in the exon region and then activate calcium activated protease, which leads to the fragmentation of the exon. So of course, they also have some other data to support this whole story. So this is just another example of how to use proteomics to answer biological questions. So I'm going to move on to a second example, also recently published by our very own Dr. Ping Ho. And Dr. Ho's lab studies pancreatic cancer, and she's especially interested in pancreatic cancer that is resistant to KRAS therapy. As you know, KRAS are mutated in many cancer, I guess more pronouncedly in pancreatic cancer. So the big story is we are able to help Ping to identify a transcriptional regulator called NFAT5 that can specifically be working together with SMAD3 and SMAD4 transcriptional regulator, and leads to the expression of certain gene product that leads to the pancreatic cancer to be resistant to KRAS therapy. How did we do it? So this is a great paper with many very, very interesting data. I'm not going to make you to go through all the data, and I can't explain all those biochemical detail. Just please focus on panel B on the right. So from Ping's analysis of phenotypical screening, they find out that in order for pancreatic cancer to evade KRAS therapy, they require SMAD4 and SMAD3, but not SMAD2. So based on that information, they were able to do an immunoprecipitation mass spec, identify binding partners against those transcriptional regulators in the pancreatic cell. And by doing this comparative analysis, they find that there are about 6 proteins binding partners that's shared between SMAD4 and 3, include one that is NFAT5. Of course, all the proteins that is also binds to SMAD2 because they know SMAD2 does not play a role in KRAS evasion. So they use this particular approach to narrow things down a bit, perform a whole bunch of downstream experiments as shown in this diagram, eventually build a very beautiful story with NFAT5. Okay, so move on. So they did, of course, for nowadays to publish in cancer translational research related studies, you have to do a lot of animal experiments. So let's focus on this 3 region that was the blue arrow. So they were able to find a drug that can inhibit NFAT5 level, as shown in panel A. And by treating the animal with this NFAT targeting drug, they were able to, let's see the panel J here. The overall survival of the animal is increased because by inhibiting NFAT5, the cancer treatment will become more effective. And of course, let's look at panel P here. The tumor size is also reduced when you inhibit NFAT5 in the animal model of pancreatic cancer. Of course, if you have any question about this, please reach out to Pimpin. So ultimately, so our proteomics data provide Pimpin a hook, and she was able to carry out substantial amount of very high-level work, eventually develop a hypothesis. So the hypothesis is how pancreatic cancer can evade KRAS therapy is because the inflammation from the pancreatic cancer leads to the secretion of TGF-beta. TGF-beta binding to its receptor can activate SMAD3 and 4, and SMAD3 and 4 can recruit NFAT, then create this multi-protein complex in the transcriptional, at the transcriptional level, leads to the elevation of S100A4. And S100A4 can be secreted from the cancer cell, which leads to pro-survival cancer KRAS treatment resistance. That's to the degree I can understand her work. All right. So I show this not only because Pimpin is a colleague, almost next to my office, but also she did a lot of work afterwards. So what we find from people doing proteomics is people have done many work at many proteins. They do not have the resource necessarily or the desire to do a lot of follow-on work. But if you do spend time carefully evaluating the data, design proper control, you can narrow things down, allow you to build a beautiful story like this. All right, so with the consideration of time, I'm going to move a little faster. So I'm sure many of you already know about proteomics. So what about multiomics that in my seminar title? So the technology that we are currently using right now is called a thermal Lumos instrument. We acquired it in 2018 with an S10 grant. And this is the one that can, Tom is able to give a lot of data for many people in the audience here. And just recent, in last year, Dr. Gauss recruited Dr. Tanya Wang to Rutgers. And as a part of her startup, she was able to buy this instrument called the DAISY mass spec. And Dr. Gauss says, Hung-Lee, you're a mass spec person, we want you to run it. So we have never run mass spec imaging, but we are learning and we are trying to set this up as a service to support everybody's research. Okay, so this instrument can allow us to image drugs, lipids, and metabolites. And also in the Cancer Center, recently Dr. Gauss was able to acquire technology called a VCM RNA spatial imaging, as well as Akoya protein imaging technology. They will be located upstairs in the Luke's histology lab. So in other words, we can do a spatial multiomics by analyzing metabolites, proteins, mRNA, all in the Cancer Center building. So there's a lot of opportunity for advancing your research. And just this year, we got another S10 grant funding to purchase a Bruker timsTOF mass spec instrument. As you compare timsTOF with the Orbitrap, it can allow you to identify and quantify twice the number of proteins, maybe even in half of the time. So potentially we can help you to get more data with hopefully cheaper costs, but that we have to wait and see. So how does this DAISY mass spec imaging works? It's a machine that allows you to put a glass tissue slides, and then the mass spec can spray charged liquid like methanol and water onto the tissue, and then that can vaporize the metabolite on the tissue, and the vaporized molecule can then be sucked in by the vacuum in the mass spec, and then allow you to generate a map of the molecule. Okay. And why is this very useful? Because first of all, it's a very easy technology. You just have to provide, for the most part, fresh frozen tissue slide. And without any other treatment, we can acquire image for you without many other complicated treatments, such as matrix application. And for each location of the tissue, we can obtain a spectra like that. you can see that many molecules can be found in tissue. And then by targeting the particular analyte of their interest, we can say display the distribution of this molecule with the size of 401. You're able to see where in the brain this particular molecule is enriched, where it's not. And this slide was provided by Ariful Islam. Hopefully he will come and join us early next year. And I'm just going to showcase Tanya Wang's. If you haven't gone to her recruitment talk last year, I'm just going to try to represent her work as much as possible. As you know, Tanya studies the host bacterial interaction, how the metabolism of the bacteria can affect the immunity of the host. And based on her analysis, first of all, they did a metabolomics analysis, identify a panel of molecules that are enriched in particular strain of a bacteria. So this is done by LCMS. You can do the same thing with Xiaoyang Su in New Brunswick at the metabolomics facility. Once she has a panel of a molecule that is changed between different bacterial species, then she did a Daisy mass spec analysis. As you can see that in one strain of the bacteria, they see the accumulation of some of those molecules identified on the left. And in other strains, there's no appearances of those molecules. Then they were able to correlate the presence of those molecules with the biological phenotype she's trying to build. And of course, you can also use this as a display to study drug treatment by adding different drugs to see whether the disappearance of these metabolites correlate with the phenotype that you are studying. So this is her example published in Cell Metabolism. So Ariful Islam has also done many other works. Here's an example showing lipid metabolites in certain animal model in different part of the brain. You can see it's very powerful. The slice of whole brain, you're able to see two arachidonial glycerol in different brain region. And so as you can see that this is the spectra from one of the brain region. And the molecule of their interest is actually relatively low. So this is the orange molecules are the ones he was interested in, and then a lot of other molecules is not. So the mass spec sensitivity allows us to quantify the spatial distribution of very low-level metabolites. Can be very useful for very low-level biomolecules. So in this next slides, he studies the supplement of different fish oil, different seed extracts to see the presence of the DHA. I don't even know how to pronounce that word. So DHA distribution in the brain as a result of the supplement. So supplement allow you to have essential fatty acid, and then they're studying by eating certain supplements, they see the accumulation of essential fatty acid in the brain to help with memory. And okay, so this again is the same thing by analyzing the phosphatidylcholine derivative of DHA. So not only the direct metabolite from the food, but also downstream metabolites that can play a role in biological function. And of course, this technology can also be used to quantify drugs. Okay, this is very useful if you are studying translational research. You want to make sure the drug you develop go into the precise tissue you intend the drugs to be. So this technology can allow you to do that. And they did something even more great. They're able to slice the whole animal, mice, and they're able to see drug distribution in the different kidney area of the mouth. Okay, of course, this will take 2 days to acquire the data, but it's doable. So I was hoping the PHI people would be there. I just want to show you the slide from Veronique Detroit, who used to be here at PHI. She also used similar technology called MALDI imaging when she was here and studied drugs that are related to TB. Okay. So in other words, right now you can do something similar to what Veronique was able to do before she went to Hackensack. Ultimately, hopefully the technology will allow you to tell a story with pictures rather than numbers. Because apparently with pictures showing changes of different metabolites, you can create a better impression with grant reviewers or manuscript reviewers. So perhaps we'll help you to get more grant funding as well as publish in higher impact journals. So how do you access it? So hopefully with all the visa paperwork being processed, Dr. Islam can arrive at Rutgers next spring and then can help set up the instrument. And then we then can start doing user training as well as take samples from different users. I just want to emphasize that I'm able to, so for sample prep, you can use the cryotome from Luke's lab, the histology lab right upstairs from us. And the data analysis, I've been in contact with Dr. Lamens and Dr. Johnson. They're willing to help you with data analysis because with imaging, we have almost no expertise. So we rely on other people with expertise to help you make sense of those data. All right, so with the last 15 minutes, I just want to go quickly for the second new instrument so it's called a Bruker timsTOF mass spec, and we just got this funded in September. It's over $1 million. And it's this box on the right, okay? So this is an instrument that can do something called ion mobility analysis, as I mentioned earlier, separation of molecules by shape. And I just want to thank the participating investigators for this grant. Without your participation, without your NIH-funded grant, we would not be able to get this instrument funded. And what's the benefit of this Ultra 2 instrument? This instrument can allow us to quantify proteins from as low as 50 picogram to as high as 1 microgram. Why do we care about this kind of analysis? For a lot of low-level analysis, a lot of protein yield is very low. And in our traditional mass spec, we cannot even see the proteins. On the other hand, why do we care about high-level proteins? Because if you give me a protein band, a mouse stem, we don't have any idea how much it is. So if we overload the traditional HPR mass spec, we may get less signal because of the oversaturation. So this new instrument allows to analyze things from very low to very high level. So one of the applications on the show is called immunopeptidomics on this second instrument. So for this instrument, many researchers want to develop immunotherapy or cell therapy for cancer, personalized therapy for cancer. So immunopeptidomics is a technology allowing you to identify antigens presented on the cell surface and in individual person, allow us to identify specifically enriched antigen. Perhaps you can use that to develop more targeted immunotherapy. And this is done in conjunction with next-gen sequencing. So we can match the mass spec data with the mutations happen in individual patient gene sequence. So how is this done? So the reason why this works is because in cancer cell, cancer proteins sometimes can be degraded by proteasome, generate peptide fragments. Those peptide fragments can then bind to HLA protein molecule, present this red cancer antigen to the cell surface. Some of those cancer-derived antigens can trigger more stronger immune response than others, right? So by collaborating with Dr. Kyle Payne at CINJ, we compared to do immunopeptidomics with our existing instrument versus the timsTOF instrument. And so Kyle's lab, they isolate the HLA protein complex and look at the peptides bound to those protein complex. And then Tom then take over the sample, perform LC-MS analysis. On our current instrument, we can find up to about 150 peptides per sample category. On the other hand, when we analyze the same sample on the timsTOF, we're able to identify 10 times more peptides. This is actually the first try without any optimization. So suggesting that the new instrument will greatly help us to perform low-level proteomics analysis. And again, from that demo analysis, Dr. Payne's lab compared 3 different conditions: vehicle, positive control with the antigen they know, as well as a new drug. And then by comparing the data from Lumos with the new timsTOF instrument, they're able to identify 10 times more peptides, including the positive control antigen that they know existed. So suggesting that this new instrument could be very useful for cancer immunology studies. And of course, we can do a volcano plot analysis showing you what peptides are specifically enriched in particular drug treatment, and then what kind of precursor proteins that those peptides are derived from, allow you to potentially use this information to design a novel antigen can make your cancer immunology therapy more effective. And another piece of data is to show you we can identify more proteins, especially at low level. In this experiment, we mixed about anywhere between 20 and 80 nanograms of E. coli protein in the background, 200 nanograms of HeLa protein. So the red line represents the proteins identified from E. coli. As you can see that with our existing instrument, we can probably find about 50 proteins at the very low level. With the timsTOF, we can probably find about 3 to 5 times more. Okay. At high level, 200 nanograms of HeLa protein, we can consistently identify about 6,000 proteins with HeLa, but about 50% more with the timsTOF Ultra, suggesting at both low end and the high end protein amount, the new instrument can allow you to get more useful data for your research. This timsTOF can also allow us to identify more post-translationally modified proteins, both for example, here, phosphopeptides and acetylation. So the aim is to install this instrument early next year and then start to be able to take sample to help you advance your research. So to summarize, right now at Rutgers, we already have enough technology, not only my lab, but also the genomics core, histology core. You can already do multi-omics analysis in bulk, as well as for some of the technology, you can do single cell analysis. But with the addition of the DAISY mass spec, we can do spatial multiomics, meaning that with the DAISY mass spec, we can help you to obtain spatial distribution of metabolites. And then working with Luke's lab, you can probably do the Akoya protein distribution. I believe he has a technology can analyze up to 30 proteins sequentially. And then there's recently this addition of the Visium RNA spatial omics platform. I believe will also be localized in Luke's lab, allow you to quantify RNA distribution on a spatial level. So takeaway is you are maybe early next year, you're able to use DAISY mass spec to analyze metabolite distribution, use the Akoya technology as well as Visium technology in Luke's lab to analyze proteins and RNA, and hopefully those multi-omics information can allow you to develop genetically newer hypotheses, help you to compete successfully for your grant application. And the DAISY mass spec imaging is very useful for studying tissue distribution of drugs and biomolecules. The new timsTOF mass spec can allow you to get more information with less sample. And ion mobility on both the DAISY mass spec as well as timsTOF mass spec can allow you to resolve some of the complexity of the molecules that share identical mass. Okay, so before I make an acknowledgement, I just want to make a public service announcement, not just for my core, but for other cores. So it is very critical that when you obtain data from people from our core or from other core, when we ask you to acknowledge our S10 grant, of course, it's beneficial for us because then we can tell Dr. Gauss how much this new instrument can support you. But also, NIH directly asked us to convey to you that your acknowledgement of the S10 grant will help them to ask more money from the federal government, especially in light of the election last night. So we need more justification for NIH to get money to funnel down the research. But if you don't acknowledge us or acknowledge the S10 grant, NIH has no documentation. So it's important. It's important for your grant. It's also important for us as well. And also another gray area is the when to put core facility people in acknowledgement, when to put us co-authorship. So many of you may think, oh, buying service from core facility is like buying antibody from Sigma, but it's different. So Association of Biomedical Research Facility actually have a guideline. So if you have time, you can go to read the guideline. Essentially that, sorry. Essentially for core facility people, we also, many of us are PhDs, we are scientists. When we make contribution intellectually to your experimental design and data analysis, the core personnel people deserve to be acknowledged just as other co-author. And also from a selfish point of view, some of the core facility people, in order to keep them, they have to see their career advancement, have something to document. So we need to be able to keep experienced people, make them happy. And then also the existence of core facility depends on, in part, on your acknowledgement in papers or publications. Many of you, when you obtain data, publish your own paper because we didn't make intellectual contribution, that's okay. But we have no idea you actually published the paper with our data. So we can't tell Dr. Gauss to say we have, different people. So please let us know. And of course, for routine data analysis, there's no need to put us in the paper. The difficult part is what is routine, what is not. So try to clarify, if we participate in, have a lot of discussion with you in design the experiment, read the paper you give us, we spend a lot of time in helping you to design the experiment, that's intellectual contribution. And of course, if you have submitted the manuscript, review, ask a lot of questions that require us to clarify for you, and then especially want us to produce more substantial other things to help your paper to get published. That's also intellectual contribution. And so ultimately, it's really up to you. But the more you can help us to document our contribution to your research, the more we can help you in the future. Because the core facility, if we don't show productivity, then the core facility may not be there for long. So that's not only for this core, but it's for every core. So with that, I want to acknowledge the NIH support for all the grant funding. In fact, we have one more grant, instrumentation grant that is pending. Hopefully it will be funded next year. And so we want to acknowledge the support from Dr. Gauss as well as Dr. Lazzarino to subsidize the core. And then we want to thank Dr. Scotto and Dr. Omari for providing the funding. Whenever we write S10, they have to provide institutional support to help subsidize the service contract so we can lower our fee to encourage the usage. And now, of course, thank Vivian to the departmental support, as well as thank Dr. Wang to bring in the DAISY mass spec, and Dr. Johnson and Laments offering to help with the imaging data analysis. So with that, I'm happy to answer any questions. Yes.
So regarding the timsTOF instrument, how it is different in terms of sensitivity, activity, and resolution compared to the Orbitrap or a high resolution one like FTICR?
It's pretty sensitive. We haven't it's just been installed, so I can't answer you that question with hands-on experience. But from Dr. Tony Wang's experience, for most applications, it's pretty enough. It's probably not as sensitive as triple quad, but it's probably more for small molecules. It's probably more sensitive than some other models. Because it's a QTOF instrument, so it does have high resolution, also have ion mobility. Speed, typical image probably takes about 4 to 5 hours for a quant.
No, not a bit. I would ask selectivity, like if they want to distinguish two molecules that are so close.
Then you have to do MSMS.
Oh.
Yeah. Question? Yeah, Gilbert?
I noticed you give a drug and then you map it. Doesn't this drug have to be tagged?
No, each drug has a mass. So we're actually measuring the size of the drug itself. That's the beauty of this very little sample prep. Provided your drug is not broken down by liver into some other species.
Oh.
Yeah. Yes, David.
Do peptides with d-amino acids, a mixture, show a different signal, or is it the same molecular weight?
I could put that's the same molecular weight because it's still the same amino acid, but it could show different ion mobility. Means it could change the shape of the
Has
peptide.
that been shown with respect to bacterial cell wall?
I haven't found a paper to tell you the answer, but I imagine yes. Yeah. Yes.
So you talked a little bit about some of the different analysis modalities that you have for the DIA analysis. Can this detect, is it sensitive enough to detect non-canonical modifications to your peptides and proteins like C-terminal amidation or lipidation or other small changes?
Yeah, with the resolution of you are talking about peptides, right?
Yes.
Yes, but by peptide level, probably not at protein level.
Sure.
But you have to know what you're looking for.
Okay, that's the caveat.
Yeah, you have to, because the software looking for changes in mass, if you don't tell us what you're looking for, the software cannot look for a mass change. Yes, I don't know whether people have questions.
Just ask to shout out, ask them to shout out.
But we
Anybody?
can't hear.
Usually you can.
Where is Oriana?
Or ask people to put it in the chat.
Okay, so for the people online, please, there are 2 questions on chat. Oh. Okay, that's all. So if you have any questions, please put in the chat, otherwise we can wrap up.
Thank you.
All right, so no questions. Thank you so much.